Welcome to the SWAM NPTEL course on Food Microbiology for Safe and Sustainable Food Systems. We were discussing diseases caused by food-borne microbes. Now we will be discussing food intoxication and infections. And we will be covering the introduction, bacterial food-borne intoxication like Bacillus cereus, which is also known as emetic poisoning, Staphylococcus aureus or staphylococcal poisoning, Clostridium botulinum or botulism, then mold intoxication, major types of mycotoxins and mushroom toxication. Food-borne intoxication is caused by consumption of food contaminating toxins previously produced by microorganisms. Toxins cause food-borne diseases such as gastrointestinal and systemic disorders. Viable microorganisms need not be present in the food at the point of consumption for food-borne intoxication. The following are some of the general characteristics of this food poisoning. Toxins are produced by a pathogen while growing in a food. Ingestion of a food containing toxin is necessary for intoxication. Symptoms generally occur quickly. Symptoms differ with the type of toxin. Staphylococcal intoxication, enteric emetic poisoning of Bacillus cereus, botulism, toxigenic molds, poisonous mushrooms and biogenic amines are briefly discussed in this class. The bacterial food-borne intoxication, Bacillus cereus or emetic poisoning. Emetic toxins are produced by Bacillus cereus during growth in food. The emetic poisoning particularly associates with starchy products such as cooked rice dishes, fried rice, puddings, sauces, flour and pasta dishes. Fast food commonly associated with emetic poisoning such as reheated fried rice and wheat based products. This pathogen is a frequent contaminant of uncooked rice and their spores can survive in the cooking process. The rice is also prepared in bulk with boiling for preparation of meal. The spores are more heat resistant and may survive during cooking. Following cooking, the rice or bulk rice is kept at room temperature for a long time. This can allow the spores to germinate. The cells will then multiply in food and produce toxins within cells. Reheating rice prior to serving will not inactivate spores and toxin. The toxin is a heat stable protein. Lysed Bacillus cereus cells release emetic toxin. Usually 10 Bacillus cereus cells per gram of food are sufficient to produce enough toxin to cause illness. Emetic symptoms develop within 30 minutes to 6 hours after consumption of food containing emetic toxin. Emetic symptoms are nausea, vomiting, abdominal pain and watery diarrhea, but no fever. The symptoms last for 24 hours. In some respect, emetic symptoms are similar to staphylococcal gastroenteritis. The emetic toxin is stable at 121 degrees Celsius for 90 minutes and resistant to proteolytic enzymes and low pH. Toxin producing stain cannot grow below 10 degrees Celsius and produces more heat resistant spores. This is the description of Bacillus cereus. This is gram positive rod shaped facultative anaerobic bacterium and belongs to Bacillus group. This causes emetic and diarrheal disease. Emetic means the emetic disease is caused by consumption of cooked rice or other starchy foods left at room temperature for a prolonged time, and diarrhea is caused from the meat and ready prepared meals that were improperly stored and causes diarrhea and stomach cramps. About 10 to the power 5 to 10 to the power 8 organisms are required to initiate the disease, and the foods which are affected are rice and other starches, prepared foods, bulk milk, rice and other starches, prepared foods, milk, meat, vegetables. About 64,000 cases are reported per year in the US. This Bacillus cereus actually that causes the emetic, produces emetic toxin and which induces vomiting, whereas the diarrheal toxin that will go into the intestine and target it, and then while passing through the peristaltic movement it causes diarrhea. The Bacillus cereus is gram positive rods, endosporic, non-capsulated, motile, gastroenteritis, phospholipase C, emetic toxin, diarrheal toxin and hemolysin. How to detect this? The differential diagnosis of Bacillus cereus are the media which are used are the nutrient agar, on that the rough and dry colonies are visible, a blood agar and MYPA, that is the mannitol yolk and polymyxin agar is used. They are gram staining. If it is seen under the microscope after gram staining, then it looks like short chains of rods, and flagella staining also shows the flagella. Other tests which are performed is catalase, it is catalase positive, mannitol fermentative, methylene blue reduction test is positive, motility test is positive, whereas the string of pearl is negative. So, this by that way we can identify the Bacillus cereus, and treatment lies that do not stop vomit or diarrhea. Intravenous fluid therapy, vitamin B complex is given and minerals. Now, the Staphylococcus aureus or staphylococcal poisoning. Staphylococcal food poisoning is caused by enterotoxins that are produced by Salmonella aureus, that are produced by Staphylococcus aureus. Staphylococcal enterotoxins are heat stable enterotoxins. Staphylococcal enterotoxins are heat stable exotoxins. Staphylococcus aureus is a gram positive spherical, that is coccus, to ovoid cells, catalase positive, oxidase negative, mesophilic and facultative anaerobic bacterium. It appears as grape like clusters under microscope. It ferments mannitol, hydrolyzes gelatin and differs in sensitivity to bacteriophages. Not all Staphylococcus aureus strains produce enterotoxins. In general, enterotoxigenic Staphylococcus aureus produces catalase, thermonuclease and hemolysin and ferments glucose under anaerobic conditions. Bacteriophage typing can be used for detection and relationship between strains and their sources. Staphylococcus aureus grows at a temperature of 7 to 48 degrees Celsius with an optimum at 35 degrees Celsius. It grows at low water activity, that is up to 0.86 and with 7.5% NaCl. Growth pH ranges from 4 to 9.8 with optimum being 6.5. In order for food poisoning to occur, the food containing an enterotoxigenic Staphylococcus aureus must be held at a warm temperature, usually room temperature or above, for several hours, at least 4 hours, and must grow to sufficient numbers to produce enterotoxins. Normal cooking temperature of foods will not destroy the enterotoxins while killing Staphylococcus aureus. Alternatively, the lack of Staphylococcus aureus or the presence of low numbers does not indicate the absence of enterotoxins in the suspect food. This is especially important in the case of heat processed foods. Streptococcus aureus can produce enterotoxins at water activity more than 0.92, while bacterium grows up to water activity more than 0.86. Enterotoxin production occurs near optimum pH value, that is 6 to 7. Staphylococcus enterotoxins is resistant to proteolytic enzymes and low pH, that is up to 2, which allows it to pass through digestive tract. Disease and symptoms. The following conditions increase the risk of staphylococcal food poisoning: inadequate refrigeration, poor personal hygiene, inadequate cooking or heating of food, prolonged use of warming plates when serving foods, and holding the food at a warm temperature for several hours to grow Staphylococcus aureus to sufficient numbers and produce exotoxins and produce enterotoxin. The consumption of 10 microgram of staphylococcal enterotoxin together with foods is sufficient to cause staphylococcus food poisoning. Some staphylococcal toxins are encoded by chromosomal genes. Prevention and control. The control of staphylococcal food poisoning can be achieved by the following practices: wash hands and under nails vigorously with soap and water before handling and preparing food. Do not prepare food if nose or eye is infected. Do not prepare or serve food for others if wounds or skin infections are present on hands. Keep kitchens and food serving areas clean and sanitized. If food storage is necessary for longer than 2 hours, keep hot foods hot, that is more than 60 degrees Celsius, and cold foods cold at less than or equal to 7 degrees Celsius. Store cooked food in a wide shallow container and refrigerate as soon as possible. This is the flow diagram how the enterococcal food poisoning takes place. First, the cooked or processed food contaminated with Staphylococcus aureus, then it produces enterotoxin which is consumed, and then it will go to the stomach where the SC, that enterotoxins binds the submucosal mast cells which release 5-HT. The 5-HT then binds the 5-HT3 receptor on vagus nerve in stomach. Then 5-HT stimulates vomiting center, that is the medulla oblongata in brain, and then it induces the vomiting within 30 minutes to 8 hours with an optimum of 3 hours. The another organism is Clostridium botulinum, which causes botulism. Clostridium botulinum is gram positive motile with peritrichous flagella, obligate anaerobic, straight or slightly curved rods, produces terminal or subterminal oval endospores and occurs as a single cell or in small chains. It produces 7 types of serologically distinct botulism toxins, which are generally neurotoxins designated by the letters A through G. Single strain of Clostridium botulinum produces one type of toxin. Human botulism, including food-borne wound and infants' botulism, associates with neurotoxin types A, B, E and F. Type C and D usually associate with animals and birds. Type E commonly associates with fish. The growth pH ranges from 4.7 to 8.9 with optimum being 6.5. This is the description of different types of enterotoxins produced by botulinum. Type A, it is the one commonly causing human botulism in the western part of the United States. It is more toxic than type B. The type B, it is found more often than type A in most soils of the world, and it is less toxic in human beings. Type C, it causes botulism of fowls, cattle and other animals, but not of human beings. Type D, it is associated with forage poisoning of cattle in the Union of South Africa. Type E, it is toxic for humans and is obtained from fish and fish products. Type F, it is similar to types A and type B, has been isolated in Denmark and produces human botulism. Type G is isolated from the soil in Argentina, but has not been implicated in human botulism. Clostridium botulinum is sensitive to low pH, that is less than 4.7, low water activity, that is 0.93, and moderately high salt, that is up to 5.5% NaCl. Spores do not germinate in the presence of nitrite up to 250 ppm. Strains can either be proteolytic or non-proteolytic. Proteolytic strains can grow at 10 to 48 degrees Celsius with optimum at 35 degrees Celsius, and the non-proteolytic strains can grow at wide range of temperatures, that is 3.3 to 45 degrees Celsius with optimum at 30 degrees Celsius. Spores are highly heat resistant, that is killed at 115 degrees Celsius, but the cells are killed at moderate heat treatment, that is at pasteurization temperature. Neurotoxins are produced during growth of cells in foods. The neurotoxins are heat labile and can be destroyed at 60 degrees Celsius in 10 minutes or at boiling temperature in 5 minutes. Radiation at 5 to 7 megarad can destroy toxins. Clostridium botulinum strains are also divided into 4 groups based on physiological differences. Group 1, all type A strains and proteolytic strains of type B and F. Group 2, all type E strains and non-proteolytic strains of types B and F. Group 3, all type C and type D strains. Group 4, all type G strains. Diseases and symptoms. There are 3 types of botulism. Food-borne botulism is caused by eating foods that contain botulism toxin. Wound botulism, it is caused by toxin produced in a wound infected with Clostridium botulinum. Infant botulism is caused by consuming the spores of the botulinum bacterium, which then grow in the intestines and release toxin. All forms of botulism can be fatal. Food-borne botulism is especially dangerous because many people can be poisoned by eating a food containing toxin. Food-borne botulism. Initial symptoms of food-borne botulism appear within 18 to 36 hours after consumption of food containing preformed neurotoxins produced by Clostridium botulinum. Toxins are absorbed in the upper part of small intestine and reach to bloodstream. It binds to the nerve end at the nerve muscle junction and blocks the release of acetylcholine. The earlier symptoms are gastrointestinal and generally include abdominal pain, nausea, vomiting, diarrhea and constipation. The botulism lasts for 7 days. In severe cases, the mortality rate is usually high, a 5 to 10%. Type A usually produces a higher mortality than type B and E. The neurotoxins are the most toxic substances. Lethal dose for an adult human is 1 nanogram per kg body weight. Neurotoxins are encoded by bacterium chromosome. This picture we have discussed in the last class also. This is the symptoms of the botulism. Wound botulism. Wound botulism is a rare disease and occurs when Clostridium botulinum spores that get into an wound open wound Clostridium botulinum spores germinate and reproduce in an anaerobic environment of wound and produce neurotoxins. The incubation period of Clostridium botulinum is about 1 week and it reaches other parts of the body through bloodstream. Clostridium botulinum in wounds produce toxin to cause gas gangrene. Infant botulism. Clostridium botulinum spores are ingested by infants through food, retain viability in stomach acidity and pass intestine. They can germinate and colonize in the intestine and produce neurotoxin to cause infant botulism. Infants are most susceptible to germination of spores in their intestine. Susceptibility of infants is related to their diet and indigenous intestinal microflora. The spore cannot produce the same disease generally in individual above 1 year of age. This would be due to development of natural intestinal flora. This flora prevents germination of Clostridium botulinum spores. Both types A and B associate with infant botulism. The initial symptoms of infant botulism are weakness, inability to control the head and loss of reflex, constipation and various neurological disorders. The infants usually present a generalized weakness and a weak cry. This is the prevention of infant botulism. The infant should not be given honey, corn syrup, raw or unpasteurized milk or milk products and unwashed fruits and vegetables. Prevention and control. Botulism can be prevented by inactivating bacterium, inhibiting bacterial growth and preventing contamination of bacterium with food. Home canning should follow strict hygienic procedures during production. Because botulism toxin is destroyed under high temperatures, home canned foods should be boiled for 10 minutes before consuming to ensure safety. Commercial processors use 12D concept heat treatment. Clostridium botulinum spores are probably the most radiation resistant. Radiation dose for inactivation of spore ranges from 1 to 4.5 kg in foods. In many cases, it is impractical or undesirable to treat a food product with radiation to eliminate all Clostridium botulinum spores. As a result, most control methods focus on the inhibition of spore germination and growth and toxin production of Clostridium botulinum. Mold intoxication. Molds are heterotrophic. Molds are heterotrophic and use small nutrients by absorption. Many molds can also metabolize complex insoluble materials by secretion of extracellular enzymes outside the cell wall. Toxigenic molds produce mycotoxins as secondary metabolites that are toxic to humans. Consumption of foods containing mycotoxins causes mycotoxicosis. The basidiomycetes and ascomycetes contain species of molds producing mycotoxins. Mycotoxins are contaminated by molds with foods, animal feeds and ingredients. They can cause mycotoxicosis in human and animals. Molds producing mycotoxins mostly present in 5 genera, that is Aspergillus, Alternaria, Claviceps, Fusarium, Penicillium. Toxigenic species of molds from many other genera can also produce mycotoxins. Toxigenic and non-toxigenic mold species cannot be differentiated according to morphological characteristics. A number of factors can affect mycotoxin production by molds, including moisture, that is relative humidity, temperature, substrate, pH and competing microorganisms. Molds are aerobic and thus need air for growth and mycotoxin production. They can grow slowly at very low water activity, that is 0.65, and low temperature, that is at refrigerated temperature, and low pH, that is 3.5. Many of the mycotoxins are resistant to heat used in the processing of foods. Plant and storage problems known as non-infectious factors such as bioavailability of micronutrients and insects and other pest damages can also affect mycotoxin contamination. This is the flow chart showing the mycotoxin toxicity. First, the toxins are built up in the system, then symptoms build and intensify over time and produce brain fog, chronic fatigue, psoriasis, joint pain, etc., and it can be tested only by the urine samples. Contamination of food by mycotoxins. Toxigenic mold contamination can be divided into 3 groups according to the location of mold infestation with grains and mycotoxin production in food. Those that invade before harvest, commonly called field molds. Those that occur only after harvest, called storage molds, and those that easily attack damaged grains, called intact molds. There are 3 types of toxigenic field molds. Plant pathogens such as Fusarium graminearum, growing on stressed plants such as Fusarium moniliforme and Aspergillus flavus. Initially colonizing on the plant before harvest such as Penicillium verrucosum and Aspergillus flavus. Aspergillus and Fusarium species are likely to be most significant mycotoxin producing field molds. The toxigenic storage molds are principally Aspergillus, such as Aspergillus flavus and Aspergillus parasiticus and Penicillium. The storage molds grow and produce mycotoxins on grains and their products. The primary factors influencing mold growth in stored foods are moisture, that is water activity, and temperature. Intact molds normally do not contaminate grains and other products, but easily attack damaged ones and require high moisture content for growth. The examples of this group are Aspergillus clavatus, Aspergillus fumigatus, Chaetomium, Absidia, Mucor, Rhizopus and Scopulariopsis. The major foods susceptible to growth of molds and mycotoxin production include cereal grains such as corn, wheat, barley, peanuts, rye and rice, figs, dried fruits, beans, cheeses, sausages, apple cider, grain meals, cotton seeds, spaghetti, spices, pepper, unrefined vegetable oils, edible seeds, oil seeds such as peanut, soybean, cottonseed and sunflower, and various tree nuts such as pistachio, almonds and walnuts. Spores of molds exist in the air and soil throughout the world and can germinate when appropriate conditions arise. Mycotoxins appear in food chain as a result of mold growth. Even if the mold contaminated food is not eaten, by humans, the mycotoxin is still dangerous to human health because foods such as crop products may be given as animal feed to farm animals. Feeding moldy products to food animals can contaminate foods of animal origin such as milk and egg with mycotoxins indirectly. Mycotoxins can also contaminate the ingredients of foods. Most specifically, the presence of mycotoxin in foods depends on several factors. Biological factors which include crop susceptibility and mold compatibility. Environmental factors include temperature, moisture, mechanical injury and insect damage. Harvesting factors include crop maturity, temperature and moisture, and the storage factors include temperature and moisture, and distribution or processing factors include the temperature and moisture. Consumption of mycotoxin contaminated foods is the main route of exposure of humans to mycotoxins. Major types of mycotoxins. The first is aflatoxins. Aflatoxins are well absorbed orally, while exposure to human skin results in slow absorption. Aflatoxins are toxigenic, mutagenic, hepatotoxigenic and carcinogenic. They are produced by Aspergillus flavus, Aspergillus parasiticus and Aspergillus nomeus. The aflatoxin contamination is a problem of poor processing and storage conditions of cereals, allowing the growth of molds. High humidity and warm temperature can raise the level of aflatoxin in food. There are aflatoxin B1, B2, G1 and G2, while M1 and M2 are metabolic products of animal systems. M1 and M2 are metabolic products of B1 and B2 respectively in milk. Aflatoxin B1 is the most toxic of the group, and toxicity of aflatoxins decreases in the following order, like B1 is the most toxic poison, followed by M1, followed by G1, followed by B2, followed by M2. M2 and G2 have equal toxicity. Aflatoxins can lead to liver and immune system problems. The combination of hepatitis B infection and eating foods containing aflatoxins highly increase the risk of liver damage. Aflatoxins produce acute liver damage with cirrhosis, carcinogenic and hemorrhagic effects, altering digestive tract and immunosuppressing. Aflatoxins can suppress DNA and RNA synthesis. This leads to structural changes in the cell and reduction of protein synthesis. Aflatoxins inhibit oxygen uptake in tissues by acting on the electron transport chain, inhibit various enzymes and decrease production of ATP. Now, the alternarial toxins. Several species of alternaria such as Alternaria alternata, Alternaria citrei, Alternaria solani and Alternaria tenuissima produce different mycotoxins in apples, tomatoes, wheat, barley, pears, citrus fruits, peppers, potatoes and blueberries. Alternaria toxins are alternariol tenotoxin, alternariol monomethyl ether, altinuene, tenuazoic acid and altertoxin. Alternaria species require high moisture conditions for growth and toxin production. Alternaria species require high moisture conditions for growth and toxin production. Post harvest occurrence of Alternaria species in fruits and vegetables is more common because moisture content remains high after harvest. Now, citrinine. Citrinine is a significant renal toxin. It is a yellow colored compound and produced by Aspergillus ferius, Penicillium citrinum, Penicillium expansum and Penicillium verrucosum. It can occur in cereals, that is rice, wheat, oats, barley and rye, moldy bread and cheese. It can cause watery diarrhea, heart distress, cardiac beriberi, labored breathing, nausea and vomiting, and reduces weight gain due to kidney damage, that is nephrotoxins. Ergotism. Ergotism is a disease and associates with the ingestion of cereals contaminated with Claviceps species. It is caused by Claviceps fusiformis, Claviceps paspalii and Claviceps purpurea. Claviceps species are specialized parasites of small grains like rye and barley. Ingestion of cereals and cereals derived products such as rye bread containing ergot can cause serious symptoms. This mold infects flavors of grasses and cereals. The tissues of infected grains are replaced by mold mycelium to produce purple brown toxicolates, ergot, toxic alkaloid ergot, that is ergotamine. Ergots may be incorporated into flour and bread made from grains. Ergot alkaloids cause hallucinogen, convulsions and gangrene. Fumonisins, Fusarium verticilloides is one of the most prevalent fusaricin producing molds associated with corn across the world. Fumonisins are also produced by Fusarium proliferatum and another Fusarium species. They can grow on agricultural commodities in the field and during storage. Approximately 20 fusaricins are characterized. Of these, fumonicin B1, fumonicin B2 and fumonicin B3 are major natural fumonisins. The most prevalent and toxic fumonicin in corn is FB1. FB1 can cause cardiovascular depression, renal and liver lesions, and cancer of liver, oral cavity, cancer of liver, oral cavity, pharynx and esophagus. FDA sets level of fumonisins in human foods as 2 ppm. Now, ochratoxin. Ochratoxins A and ochratoxin B are potent nephrotoxins and produced by Aspergillus ochraceus, Penicillium cyclopeum, Penicillium expansum, Penicillium variable and Penicillium verrucosum. Ochratoxins cause kidney damage. For example, Balkan epidemic nephropathy reduces kidney size and fibrosis and carcinogens. They can occur in direct cocoa beans. They can occur in dried cocoa beans, oats, dried fruits, citrus fruit, peanuts, green coffee beans, corn, nut, soybean, cheese, sausage, peppers, rye, cured meats, barley, wheat and milk. They contaminate animal feed, so meat and milk can be contaminated with ochratoxin indirectly. Now, potulin. Potulin is a lactone potential carcinogen and teratogen, and causes neurological and gastrointestinal disorders. It is most commonly produced by Penicillium expansum in fruit juices and rotting apples. It is also produced by Aspergillus clavatus, Aspergillus ferius, Penicillium cyclopeum, Penicillium claviforme, Penicillium potulum, and Bysoclemys nevea. It also occurs in other fruits such as pineapples, bananas, pears, grapes, and peaches, and grains, moldy bread, cider, cheese, raw and cooked meats, cured meats, sausages, and nuts. Molds can be killed by heat, but potulin is not inactivated. Quality and safety of food depends on careful handling through the whole production process. Pasteurization of fruit juices should be done at 70 to 90 degrees Celsius for several minutes. Under UV light, lemon yellow fluorescence develops. Now, the mushroom toxication. There are about 10,000 kinds of mushrooms, but only about 100 kinds are considered edible. Poisonous mushrooms produce natural toxins such as azamanitine, gyrometrin, orellanine, mucirine, ibotenic acid, mucimol, psilocybin, and coprin. Mushroom poisoning is caused by the consumption of both raw and cooked mushrooms. Most mushroom toxins cannot be made non-toxic by cooking, canning, freezing, or any other processing. Thus, the only way to avoid poisoning is to avoid consumption of toxic mushrooms. These are the examples of some toxicity like cholinogenic toxicity. It is produced by clitocybidiol beta, and the toxin produced is mucarine, and the antidote is also available, that is atropine and glycopyrrolate. Whereas the disulfiram like toxicity is produced by Coprinus ultramentarius, and the toxin is coprin. There is no antidote. Hallucinogenic, it is produced by Psilocybe cubensis, and the toxin name is psilocybin psilocin. For this also no antidote is available. Isozazole syndrome, it is produced by Amanita mucaria, and the toxin is ibotenic acid and mucimol. For this also no antidote is available. Mushroom poisoning is generally acute with different symptoms. There are 4 groups of mushroom toxins. Protoplasmic poisons, the poisons that result in destruction of cells followed by organ failure. The neurotoxins are the poisons that cause neurological symptoms such as profuse sweating, coma, convulsions, hallucinations, excitement, depression and spastic colon. Gastrointestinal irritants are the poisons that produce rapid and transient nausea, vomiting, abdominal cramping and diarrhea. And the last one is disulfiram like toxins are the poisons that interfaces with the breakdown of alcohol after consumption of alcoholic beverages, and mushroom will cause headache, nausea and vomiting.